ahead and get started with the demonstration. Today's session is a product demonstration. It is an introductory demonstration to Pristima. Pristima is a total preclinical management solution that combines both software, services, and best practices. The Pristima Application Suite is the most comprehensive end-to-end preclinical enterprise solution on the market today. It's unique in that it combines both research data management as well as animal management through Pristima and the Pristima VM Suite. Pristima covers a broad spectrum of features and functionality, from subject acquisition or creation through health and welfare functions, research study design, the actual conducting of the research and collection of data, finally to the analysis of the data and the reporting. Today's session is going to be presented by Tom Klapmutz. Tom is the Senior Customer Solutions Manager at Xybion Pristima. He brings over 20 years of experience in discovery, preclinical safety assessment, and laboratory animal resources. Prior to joining, he was a senior scientist and supervisor at Merck Research Labs, formerly Schering-Plough Research Institute. He has a lot of experience in SP development and the implementation of toxicology study protocol, template creation, and procedural development, and was involved in the validation of Schering-Plough's data collection system. He's a member of the American Association of Laboratory Science and certified as a laboratory animal technologist. With that, I want to introduce Tom, and Tom, thank you again for taking some time out to spend with us today and show us a little bit about Pristima. I'm going to go ahead and make you the presenter.
Okay.
And you should see that pop up on your screen, and we'll verify that we can see your screen. And we can. That's coming through great. So I'm going to pass the audio controls over to you, and you have control of the screen, Tom, and you can take it away.
Okay, great. Welcome, everybody, to today's demonstration. And like Kevin said, we're going to go through a high overview of all the system functionality. Really going to focus on the main modules, which are colony management, study management, clinical pathology, pharmacy, and then necropsy histology. So what I wanted to do is, this is the main screen, the main administrative screen, where we can also manage security, create animal species strains, create measurements. With Pristima, there are your basic measurements that are part of the system: body weights, dosing, clin signs, food consumption, palpable mass, necropsy, tissue collection. But then Pristima also has the ability for you to create measurements for whatever data points you want to collect. And I have a couple samples of that as well in the demo. We have tissue management, which is where you create your tissues for histology, necropsy. Lexicon management for clin signs, fetal pathology. Facility management, where you can set up your equipment, your balances, other equipment that you may use, microchip readers. So it's fully integrated with all of the equipment that you use on a day-to-day basis in your laboratory. We have the pharmacy area, reporting, and then study login. So what I'll do is go ahead and start us off at the beginning, which is colony management. So typically when you receive your animals, they'll come in through a colony. And what I have is I have all of the areas open on the bottom to make it a little more easier so I can spend time in each of the functional areas. So just at our colony management, here's where you would set up your orders of animals. You would receive shipments of animals. And again, you have all your information. You have your completed order, so it tells you what came in on that order, your strain. You can assign it to various subgroups within the colony. The colony is managed by a protocol. So we just looked at a protocol here. And again, the same functionality is shared throughout most of these modules. So this protocol setup for colony is very similar to the study setup for an actual data collection study. So we have personnel, your phases, your schedule, and then your animal specifications for the colony. We have some definitions where you define various things within the colony protocol. We have some colony tables that are specific to the colony area. We can register substances within the colony. So within colony, there are some unique functions, such as vaccinations, treatments, supplements. And here's where you manage all of the various substances that you would administer to your animals, either as a vaccine, an enrichment, a supplement, a treatment. And then we have a dashboard, which the dashboard is an area where you can manage your colony. So you can set on a specific group of animals the measurements that you want to achieve before those animals would be available for study. So for instance, if you had a quarantine period during that time frame, you wanted to do at least 2 body weights and 5 clinical observations and 2 dewormings, you would set that information here. So until this criteria is achieved, the animals will not be available or not be able to be set to available for a study. So they're not ready for a study until this is achieved. And then in the dashboard, what the dashboard is, it's kind of a scheduling system for your colony, and it shows the colony animals, it shows their current status, it shows the measurements that are scheduled and the dates, and then you can add additional scheduled items. So for instance, one of these animals, say this animal, was sick, we can go ahead and assign a treatment, and we're going to do a deworming. We select that, it now overrides the schedule and allows me to pick individual dates that I want to do deworming for this specific animal. So it allows me to schedule only this animal that's in the colony for deworming. One of the features with colonies, you also have the ability to continue to treat them within colony while they're on study. So this is a colony that I have set up for the demo. And we can look at animals that are in the colony and animals that are on study. Just give it a minute to update. The other unique feature of colony is you're able to transfer animals from the colony to a study, and then from the study back to the colony. So if we jump over to a study real quick, and again, I'll explain this. In our animal specifications, where we determine the strain and species and where we're getting the animals, these animals are coming from the colony. And so this is where we would pick the animals. What it does then is it communicates to the colony and looks for the colony that I have animals reserved for this study within. And so I've already done this to make the demo flow easier. So these animals were selected from that colony that I was working on, a colony, and all of their information comes into the study. These animals, 105, 113, were not transferred because I only needed 22. In that colony, I had 25 per sex. So they've all been transferred into the study. And now you can see on here we have the on-study animals. So these are the animals from this colony that were transferred to that study. And then we can maintain the colony schedule, so what happens is when those animals come back to colony, they'll pick right back up on this schedule. You will not do measurements such as body weights or CLIN signs in 2 areas. It keeps the study data with the study and the colony data with the colony. If we go up to our animal screen where we get some more information about our animals, we'll bring these animals up. For each of the animals, we have the information, the colony name, the site, the colony ID. If I identified a tag tattoo or an implant ID, the species, strain, sex, date received, the subgroup they're in the colony, if they're alive or dead, their transfer status, if they're on study or not, what study they're on. Are they in a treatment phase? Are they dose naive? Are they drug naive? What is their APS or IACUC number? Pain category assignment, current location, current age. If you entered that, I did not enter birth date information into this colony. And then for each animal, you have more information: disposition history, APS history, drug history, measurement history, and then transfer history. So here you can see this animal was received in evaluation status. It was moved to no restrictions. It was then transferred to the study. And here you can see it was assigned to this study. And now it gets a study animal number and a pretest number. So the animal is identified throughout the system by various numbers. With the colony, it could be a standalone functioning system. So you can manage a colony of training animals for a university setting. It could be used for livestock. It could be used for primates, breeding facilities, breeder colonies, any type of colony type management, our colony module can be used alone. So from here, we'll go ahead and exit from the colony management, and if I click on study login, we have colony, and we enter in the colony. And then here's where we collect our colony data, such as body weights, clean signs, food consumption. The colony management was the scheduling and animal management area. So we have our animal room, which is where we collect our animal data. We also have a non-animal area, which I've labeled as study data. We have our study reporting area, and then we can go back to system administration, or we can change into a different study or colony. So I'll just touch base quickly on our colony. We can go ahead and see that we have scheduled colony activities, and if we click the update, it'll bring up any available measurements. So for this day, I have animals that are scheduled for body weights, clean signs, and deworming. We move those over to the selected measurements I want to perform data collection on, and then I can click on details, and it'll show me which animals are scheduled. The gray boxes show me that I don't have that measurement scheduled. And if we scroll down, this animal, 101, is only scheduled for deworming. He doesn't have body weights or clean signs. That's a quick way to see what you have scheduled for your data collection. We click OK, and we'll go into our data collection because we're doing body weights. We select our balance. Again, all of this is configurable based on your individual business practices. Now we have our body collection screen. We have the previous body weights that were collected and the date. We would enter our new body weights from the balance. Again, we can set up range checking. So we have a lot of functions that are business process flows that you define when you set up your system. So you can have range checking, out of range controls. You can set it so that you have to force somebody to do a second weight and then accept it. If it's outside of calibration, weights taken, so you can set your calibration ranges, you can set your acceptability range. Move on to the next animal. Daily clinical observations or clean signs. You define your glossaries. So in this case, I can mark my animal normal, or I can add a finding. When you click add a finding, this is a predefined glossary that I have. And so you would just select your findings, and then you define the different modifiers and the requirements. And then there's your clean signs. We have some special features where you can mark the remaining animals normal. So if you look at all your animals and only enter findings here, then you can use the special button and mark the remaining animals normal. Again, I'm not going to spend a lot of time on the various data collection, but I just wanted to touch base. When you're exiting every session, you have a session status, so it shows what you did and what you still have to do, and you can go ahead and exit out. That's our colony data collection. Go ahead and exit this. So now I'm moving on into our study. So from our system administration, now I'm under study management. Here you define your study information, your site, your study type, your study subtype. Again, any of these selections are user definable. You can put a textual study title. You can label it as GLP. And what this does is this allows you to be able to sort the data for give me all my GLP data, give me all my non-GLP data. There's no functionality associated with the GLP button other than the ability to label the data as GLP or non. The system comes with audit trails, so pretty much any change is audit trail. Once the protocol is approved, any change after that is audit trail. So the whole system is GLP compliant. So as we move through the sections of a protocol, we have study type, animal specifications, phase selections, group specifications. Again, we can have up to 64 different dose groups. We set up our dose specifications. Here is your dose regimen, which is your actual dose administration. Here is where you set your dose, your group dosage, your group volumes. This is an integral part of the pharmacy module. So again, this can function with or without pharmacy formulations, but if you're using pharmacy formulations, the pharmacy area comes into this protocol looks for the group, the number of animals, and all of the dose information for the calculation. And again, we have our schedule. So in this study, I have body weight, cleanse signs, food consumption, palpable mass, necropsy, all of your basic measurements. And again, the measurement types are pretty much the sky's the limit there. You can define any type of measurement, any type of data form you have, we can pretty much get it into Pristima to make your system completely paperless. We'll go ahead and exit out of the protocol. And again, we go back into study login, we switch to study, and we enter into the study. And now we're into our study data collection. Now you can see a couple more tabs. We have necropsy, pathology, and then we also have study data. We'll just touch base on study data. In this area, this measurement, this area is not set up to collect data on an animal. It's set up to collect data on a study. So if I click update, some of the type of things you can have in here is husbandry. So we can look at husbandry, and I have my area that this measurement is going to be collected in is a study notebook. So I can then print all of my study notebook data. And then we have the measurement that I created. So here, bedding changed, yes or no. You can enter your bedding lot number. Did I check my cage zippers? Did I mop the floor? Yes. And then a general comment. You can add anything you want here. So we can do temperature, humidity. And again, you see it's associated with the study. It has the date, the phase of the study. It's in their study notebook, and it's AM husbandry. Click OK, and the data is collected. Session's complete, and we can exit the study data area. When we go into animal room, we just click update, and it'll bring up all of the measurements we have scheduled for today. What I'm going to do is go through and do a couple body weights and dosing real quick. To be able to show you another feature of Pristima is our timed blood collection for TK. Now, the blood collection is based on the dose time, so I have to go in and dose a few animals. So I'll go ahead and do that. Do our body weight. Then we have our dosing screen. Again, it's going to ask for a bottle ID. So if we had a accountability set up, we'll be able to scan the dose bottle that was made in pharmacy formulation. What I'm going to have to do is come back into here once we go to pharmacy. So we'll come back to this screen and we'll do the dosing. Now I'm going to go into our pharmacy module. And with pharmacy, we have an inventory system and we have a formulation system. So inventory allows you to register substances. You have containers of substances. So if we start with substances, you can register all of your different dosing compounds. You can have your methylcellulose, your test articles. Then you define containers of that substance. So here, if I pick my methylcellulose, it shows me all of the containers that I have of methylcellulose based on container type, container use, batch number, analytical samples, bulk materials. You can assign it to a study and add other information, sponsor. You can check it in, check it out. If we pick on any of these containers, we have use history, container weight history, and these weren't weighed. You can define packages, which is a container, like a box of multiple containers, and you can define what containers are in a package. So here in this red package 1, I have this substance, this container ID. It's a feed container, batch lot, and the studies it's assigned. So it allows you to inventory and move things as a package. You can receive shipments of materials. And when you receive shipments, you can enter the number of containers. You can place orders. Again, it's internal within Pristima. It doesn't go out and actually place an order at a chemical company. You can define studies that you would do within pharmacy formulations, such as homogeneity studies for concentration of content studies, and then you set up your location. Within pharmacy, we have the formulation module, which is where we actually do our mixing of substances. And so here we have a target list, which is the calculations, and I just go into target list that I started just to show you the screens for formulations. Here we define our groups that we're going to do our formulations. These are the containers. That we're going to formulate. And then we're into our formulation screen. And so here we walk through the various steps that we have for creating our substance. So if I just look at this here, we have our steps, and these are definable by each customer. So you can put in your pharmacy process in here. So what you do is you actually create a method of formulation, which are these steps over here. The system does the calculations based on the volume and the concentration, the number of live animals and their current body weight, all automatically when you set up the target list. And so here we would scan the pre-weigh container, scan the bulk weight container, and do a pre-weigh. We would scan and verify our test article, our vehicle. We do our measurements, and then we prepare it. So you can have steps such as mixing, sonicating, vortexing, and then you go ahead and dispense into dose containers. So it gives you, our batch is going to be 12 ml, and then we're going to put that into a dose container. And so that's our formulation aspect of pharmacy. We also have capsule formulations. Again, it's another type of formulation where you're actually just weighing out the powder into individual capsules. And the system is also interfaced with Mettler's Quantos system for automatic or automated capsule filling. And then here's where you define your methods. So these would be your instructions. We can have simple instructions, or you can go robust into collecting samples on the formulation container, the dose container. It's just a little snapshot of our pharmacy formulations. We can come back in and weigh and dose an animal. I'm getting that window because I'm not actually connected to a balance, so it's looking for the balance interface in my COM port. Okay, so I've entered the container ID from pharmacy, and now I can go ahead and dose my animal. And we'll go on to the next animal. Again, you can have the system set up to sequentially go through animals. You can have it set up to scan microchips. You can have it on a confirm mode or a search mode. I just wanted to dose 2 animals to be able to show you the time to bleed module. Okay, dose this one. Okay. So with the time bleeds, we go into our ClinPath area and we set up our draw list, which defines the samples. And when we click our time bleed, we can select the time points that we want to bleed, or we can select multiple. So I want to see the 15-minute and 30-minute blood collections on my screen. And now what we have here is we have the system showing me the time remaining the time remaining until we have to bleed it. So you see that the target draw time is at 11:39:51, and I have 13 minutes and 48 seconds until I bleed it. I can just double click on here, and then it brings up the information. I have my anesthesia type, my collection site, a comment field. I can enter the lot and expiration of my tube. I can put the manufacturer. My sample is being collected at room temperature. Again, it's got the time post-dose, so it's been 1 minute and 49 seconds since dose. And now I can just sit here and wait for my timer to get to 15 minutes, and then I can mark it collected. I'm just going to go ahead and collect it early, and then it marks it as collected. We also have the ability to set an acceptance range for your TK bleeds. So I just wanted to go over, this would be a complete mapping of all the animals. So as an animal is dosed, it automatically appears on this table real time. So we've gone through all of our in-life data collection. We have palpable masses, pretty much any measurement that you need for your tox or discovery type protocols. Once we have our samples collected, we would then take our samples to ClinPath. So I just want to start with data collection in our ClinPath. And again, the data collection is based on the equipment. So this is the measurements that were run on an ADVIA. And so I just wanted to show you some sample data. So we have the sample tube ID, which again we just saw when I collected that TK blood. Just assume it's not TK, but it's a hematology or serum chemistry sample. There's a sample ID that I scanned and identified the tube. That's the tube that shows up here. Now we have all of our measurements, and when we hover over each of these, it gives us the parameter name, the result value, linearity ranges if you have those set, reference ranges if you have those set, any type of flags you may set, your status, dilution factors, coded comments, result comments. It has the transmission date and time from the ADVIA. It has sequence numbering. And then you see the various codes. So these highlighted boxes showing that this is rejected. And you can see the status is marked rejected. So anything with a red box around it was rejected. We have some other various flags as well. Again, we have the ability to exclude, comment on a sample, dilute a sample, reject a sample, accept it, request a rerun. We can save it. And then once you save it, it goes for approval. So we have technician approval. You can see here pending operator approval. And then we have management or supervisor approval. We exit out of here. No. We'll go back to the main ClinPath area. And we have our analysis screen. Again, we have all of our instruments and what samples are being run on those instruments. Here's where we define our instruments. We can define control samples to run on the instruments. Samples is where we create our draw lists. And so we have our draw lists, we have our sample tracking. And so you can see I created this draw list for TK bloods. When I click on it, it shows here that this one was filled. That's the sample that I collected in TK. So you get real-time connectivity between all of the modules. So somebody in the lab bleeding an animal, somebody in ClinPath will see that these statuses will change from pending to filled. Once they're filled, then you could do sample tracking. And if I just take this number 13, take this sample number, and go to sample tracking and enter that sample number, again, this is set up for scanning the tubes. I'm hand entering. It gives us all the information, the sample tube ID, the animal, the study, the collection measurement, the session, the panels that you're going to run on it, the sex, the group, the collection information, anything that was entered in the animal room, and then you would then receive that into your ClinPath and then go ahead and do your analysis. Again, we have panels, which are the measurements that you'll perform on the samples. And so we have a full range of sample measurements for urines, bloods, pretty much any samples you can define within the system. So we've gone from colony into a study, we've made drug for the study, we've collected ClinPath samples and analyzed those samples. Now we're moving on to necropsy. And so with our necropsy, we have various measurements you can set up in necropsy: growth observations, tissue collections, organ weights. And so this is just a sample screen of our organ weight. And then we have our tissue collection. Again, you can define collection processes as well. So if you wanted to place some tissues into special stains, you can put that in here and document that you perform that process. So if your eyes are going into modified Davidsons, you can say, eyes collected as one tissue name, and the other one would be eyes placed in Davidsons, and it would be a process completed, yes. And then we have our gross observations, which are our necropsy observations. Again, this module, the necropsy, is very similar to our histopathology module. And so here we have our final phase sacrifice, all of our tissues, our findings. We have our clin signs and our masses. They would show up down here in this screen, so you'll be able to correlate them if you set correlations. You also have up here in your view, you have data viewing, so that your necropsy technician can look at any clinical observations that were recorded, any organ weights, any palp mass findings. The system goes right back into the in-life data collection module and looks for this measurement and any data associated with it for this animal. Again, we can pick our animals and we mark them dead within necropsy. Terminal body weights are performed. We go ahead and cancel out of here. And then our last module would be our histopathology. And so here we have death statuses defined and phases. So if you're just looking to do unscheduled histopath, you can select tissues. So if you only wanted to do specific tissues in the session, so say today I'm just going to do the aorta, the bone, and the brain. When I go into my data collection session, those are the only tissues that are going to show up on my list. We have the ability to do multiple animals on a screen so you can see their information, but the main focus is on this animal's data collection. We have a full range, a couple other modes. We have a matrix mode, a command mode, single animal. We have our correlations. So this animal has no entries, no clin signs, or no palp masses. You would see the masses and clin signs down here. You see other data in these fields as well, comments. We have the ability to do a tissue comment, a finding comment, an animal comment. That's pretty much just a quick overview of histopath. We also have peer review for histopathology. And then I just wanted to touch base quickly on some of our reports. So here from our colony, we have our complete animal history report, which gives you the species, strain, the studies that it's been assigned to, and then any information that was ever done to the animal, any data collections, any vaccinations, is in a chronological order for the animal. It's the entire history, any ClinPath, blood. It's more of a raw data report. So it's tracking for your IACUC and your pain categories, everything that's ever happened to this one animal within its life within your system. We also have study findings report, individual animal listings, so you have your individual histology, date of death, phase of death, death status, all of your tissues, tissue findings, the remaining tissues that were marked normal. And we have a full range range of customizable reports. With Crystal Report Editor, you can customize reports yourself, or Xybion can customize reports for you as well. And then we do have a complete repro system. It's the same module that I showed you for the animal room. It's just repro measurement. So we have individual estrus cycle, estrus staging, we have parturition, we have C-section, fetal pathology, skeletals, viscerals, just some examples of individual implantation data, and we have supporting reports for all of these various data collections. This is just a typical raw data report. So you have your study animal, your cage, your group, user, date and time it was collected. In this case, this was a back entry. So on February 29th, I collected data for February 21st. Delivery status is the actual parameter that I collected, and that's not started. And then these are the other parameters that could have been collected for this measurement. So here you see some of the other data. So we have a full range of raw data reports, individual reports, summary reports. We have built-in statistics. And that's pretty much an overview of the entire Pristima Suite, from colony through study, ClinPath, pharmacy, histology, necropsy, and histopathology. Kevin, I think that pretty much wraps it up. We covered the majority of functionality. Again, this was quick. I didn't cover it. Again, if you need any other information, turn it over to Kevin.
Thank you very much, and that concludes the demonstration. The demonstration will be made available on demand, and all those who attended today will get a link to see how you can view and access this online demonstration if you'd like to share that with other colleagues around your facility. I'd like to thank everybody for attending. Thank you, Tom, for providing us with this brief but very useful insight into the power and scope of the Pristima application. The session is now complete. We thank you again for your time.